dear students, welcome again in our normal Elimika program. Our subject is biology, and the topic you are still having at hand is gaseous exchange and respiration, and particularly we are still dealing with gaseous exchange. Can you remember what was our discussion in our last meeting? Well, of course, we were discussing about transportation of respiratory gases. And the point that we ended up discussing was transportation of oxygen or carriage of oxygen. In the transportation of oxygen, we said that this gas is normally transported by the red blood cells. The function of one of the functions of the red blood cells is to transport oxygen. Now, these red blood cells, the red blood cells contain hemoglobin. And this hemoglobin is the one which is responsible for transportation of oxygen. Hemoglobin, this is a compact molecule. It is a compact molecule made up of 4 interlocking subunits. And these interlocking subunits are identical conjugated proteins. From your basic biochemistry point of view, can you remember what a conjugated protein is? Of course, a conjugated protein is the one which consists of 2 parts. It has a pure protein portion and a non-protein portion. Say, if this is a protein molecule, say this side is a pure protein and this side is non-protein. This non-protein portion is called a prosthetic group. Prosthetic group. So for the case of hemoglobin, if this were to be hemoglobin molecule, and we have this portion here, which is a pure protein portion, this is called globin, and this non-protein prosthetic group is called, this non-protein prosthetic group is called Haem. This prosthetic group in this hemoglobin contains iron, and it is this iron which is responsible for transportation of oxygen. We have said that a single hemoglobin molecule has got 4 polypeptide chains. If each polypeptide chain is made up of globin and Haem groups, it means one hemoglobin molecule consists of 4 Haem groups. And if each Haem group is consisting of an iron atom, and this iron is responsible for transportation of oxygen, automatically one hemoglobin molecule is responsible for transporting 4 oxygen molecules. And it is this Haem group which is responsible for the red color of the blood. The adaptation that hemoglobin has in transporting respiratory gases is that it has affinity for these gases. It has an affinity for oxygen, but at the same time, this affinity may change according to the concentration, according to the partial pressure, all according to tension of the gas we have at hand, be it oxygen or carbon dioxide, as we are going to see. Now, how is this hemoglobin involved in transporting oxygen? We did say that in areas where the tension of oxygen is high, for example, in lungs, where the tension or partial pressure of oxygen is high, hemoglobin physically combines with oxygen. This iron atom physically combines with oxygen. It is not a chemical combination. It is not a chemical process. Therefore, there is no oxidation of iron by oxygen. Rather, it is a pure physical process which involves a combination of hemoglobin with, we have said that there are 4 heme groups and therefore 4 oxygen molecules. So we have 4 oxygen molecules combining with hemoglobin, then to form an intermediate product. This is called oxhemoglobin. This is oxhemoglobin. This is formed in areas where the partial pressure of oxygen is high. I have said that the affinity of hemoglobin to respiratory gases will depend on the tension of the gas that we have. Now, in areas where the partial pressure or tension of oxygen is low, for example, in the muscles, and therefore where the concentration or tension of carbon dioxide is high, the hemoglobin's love to oxygen diminishes altogether. Hemoglobin is no longer in love with the oxygen. Rather, it shunts it there and then picks up carbon dioxide. So it means hemoglobin's love to either of these 2 gases will depend on how much or how great amount of this gas is available. The greater the concentration of the gas, the increased love of hemoglobin to that particular gas. So in the muscles, where the concentration of carbon dioxide is high, and therefore the concentration of oxygen is low, oxhemoglobin dissociates into free hemoglobin and the free molecular oxygen, and then it picks up carbon dioxide. By combining with the carbon dioxide, then it forms another intermediate product. This is called the carb-amino hemoglobin. So, in areas where the partial pressure of carbon dioxide is low, for example, in muscles, we have this oxhemoglobin. There is carbon dioxide there. So it means hemoglobin will combine with carbon dioxide, and then it unloads the previously loaded oxygen. What advantage do we get by having hemoglobin behaving in this way? Having its level increased with increasing concentration of carbon dioxide, but at the same time, having its level increasing with increasing concentration of oxygen. What advantage do we gain out of this behavior that is possessed by hemoglobin? Try to think of that. But remember that, as we are going to see, oxygen, we have seen, that is combining with hemoglobin at a point where iron is. So the point of oxygen transportation is on the prosthetic group, the heme group. However, carbon dioxide, when combining with the hemoglobin, does not take the same position as that one taken by oxygen. Carbon dioxide is not loaded on hemoglobin by iron atom. Rather, it is loaded by hydrogen, it is loaded by hemoglobin on the amino group of the globin portion. That's why the product is called carb amino hemoglobin. Carb amino hemoglobin. Well, my dear student, you've been forced to review this in details because last time it is a point that we concluded with. And according to the reactions that I received from you, it seems that this portion was not clearly understood in our last lesson. Therefore, it's my sincere hope that by repeating it in details, by repeating it slowly, that it has now been clearly understood. The point which hasn't been understood yet, our numbers are there, our means of communication is there, as we have been communicating, as we have been delivering answers and discussions to you. Yes, now, having seen the transportation of oxygen, now let us see the concept of oxygen, hemoglobin, dissociation curve. The oxygen hemoglobin dissociation curve. Well, the amount of oxygen that combines with hemoglobin is determined by its percentage saturation, the percentage saturation of oxygen, or the amount of oxygen which is to combine with hemoglobin is determined by the percentage saturation of hemoglobin with oxygen. Now, when this percentage saturation is plotted against the tension or partial pressure of oxygen, an S-shaped curve is obtained. And this curve is what we call oxygen dissociation curve. That is, you plot percentage saturation against the partial pressure of oxygen, maybe in kilopascals. What we are getting is an S-shaped curve. This is called oxygen dissociation curve. Say we have 0, 100, and maybe this is 95. This is a loading tension of hemoglobin. Here it means is where hemoglobin combines with oxygen to form oxhemoglobin. Here we have unloading tension. It means it is at this tension where this oxhemoglobin dissociates into free hemoglobin and molecular oxygen. Well, you can see that hemoglobin loads oxygen at this particular tension or partial pressure. But when the partial pressure is as low as maybe 6 kilopascals, this hemoglobin will unload. It means when the tension is low here for oxygen, it will be high for carbon dioxide. So this hemoglobin will dissociate, the oxyhemoglobin will dissociate to release oxygen so as to combine with the carbon dioxide. And when the partial pressure is as high as maybe 12 kilopascals, this hemoglobin will be at liberty to pick up oxygen so as to form oxyhemoglobin. Now a question, call it a food for thought. Call it a food for thought. Why this curve is an S-shaped? Why the oxygen dissociation curve why S-shaped? Why S-shaped? Go through various books, different sources, and then you come out with an answer. I'm expecting to get your reactions by using our normal platform where we use it to meet. We have that WhatsApp group. We have our Instagram page. I'm expecting to see your reactions. Now, we have seen this oxygen dissociation curve. Now, let's look at the factors. We have the factors influencing the oxygen hemoglobin binding affinity. Factors influencing oxygen hemoglobin binding affinity. We have 3 main factors that influence the oxygen hemoglobin binding affinity. The first one, we have what we call Bohr effect. The second one, we have temperature. And the third one, we have body size. So, to start with, let us look at the Bohr effect. We have seen that the affinity of hemoglobin for oxygen decreases with an increase in the partial pressure, with an increase in tension, with an increase in the concentration of carbon dioxide. And therefore, in those body parts where the concentration of carbon dioxide is high, for example, in the actively respiring muscle cells, carbon dioxide will combine with hemoglobin to form carb-amino hemoglobin. And these areas, it means the affinity of hemoglobin for carbon dioxide is higher than that for oxygen. And therefore, in these situations, the oxygen dissociation curve will shift towards this particular side. It shifts towards this side. It shifts towards the partial pressure axis. It is shifting towards the partial pressure axis, meaning that this is an oxygen dissociation curve. I mean, with the increased partial pressure of carbon dioxide, the oxygen dissociation curve will be displaced to this particular side. With further increased partial pressure of carbon dioxide, the oxygen dissociation curve is again displaced to the partial pressure of oxygen axis, to the axis of this oxygen partial pressure. Now this movement of the oxygen dissociation curve towards this particular side is what we call a ball shift. This is called a ball shift. And therefore, what is the significance of this? Of course, we have, with increased concentration of carbon dioxide, it means there are increased chances for the oxygen to be released into the respiring tissues. This means that with the increased rate of respiration, there is an increase in the production of carbon dioxide. And therefore, with the increased production of carbon dioxide, it means its tension is also increasing. With the increased tension of carbon dioxide, automatically there are increased chances for the hemoglobin to release oxygen into the actively respiring cells. And this phenomenon is what we call Bohr effect. That phenomenon is what we call Bohr effect. Now, suppose one wants you to explain what is Bohr effect and why does carbon dioxide results into this Bohr effect. How is carbon dioxide capable of bringing about this effect? Of course, this is because carbon dioxide, when dissolved in water, it forms weak carbonic acid. That is, this carbon dioxide dissolves in water to form weak carbonic acid. And this carbonic acid, in effect, dissociates to form hydrogen ions and bicarbonate ions. It forms hydrogen and bicarbonate ions. They formed hydrogen ions. Then combine with hemoglobin, and by combining with hemoglobin, they make hemoglobin less likely to carry oxygen. That is how this Bohr effect is brought about. Now, the second factor, which is influencing the oxygen hemoglobin binding affinity, is temperature. That is temperature. Normally, increased metabolic activity results into increased temperature in a given part of the body. If you refer to your Form 3 regulation knowledge, you remember that when the environment is overcooled, one of the responses that a body indicates it to increase the rate of metabolism. And this increased rate of metabolism aims at generating heat. With generating heat, it means temperature is increasing, and the aim is to make sure that these endotherms, these homoiotherms, have their body temperatures kept constant. Well, with the increasing temperature, there is an increase in rate of metabolism. With the increasing rate of metabolism, automatically there will be production of carbon dioxide. And when this carbon dioxide is produced, it results, with the increased tension of carbon dioxide, it means hemoglobin now releases or unloads oxygen into the highly metabolic metabolic cells and picking up carbon dioxide. In these situations, the oxygen dissociation curve shifts. The oxygen dissociation curve is displaced towards the oxygen partial pressure axis. That is, we have the oxygen dissociation curve, and this is the partial pressure axis. So with increasing temperature, it means because of carbon dioxide production, there will be shifting of the oxygen dissociation curve. There will be shifting of the oxygen dissociation curve. Suppose that maybe the oxygen dissociation curve was at a particular position when the temperature was exactly 37 degrees centigrade. When it became maybe 37.3 degrees centigrade, we have the oxygen dissociation curve slightly displaced towards the oxygen partial pressure axis. And with maybe 37.5, here is where the oxygen dissociation curve is. Now, in all these cases, whether it is Bohr effect or it is temperature, and we are going to see body size, the concept is locked on the production of carbon dioxide. With increasing temperature, the rate of metabolism is high. The higher the rate of metabolism, the higher will be the tension of carbon dioxide. The higher will be the amount of carbon dioxide so generated. And with the increasing concentration of carbon dioxide, it means hemoglobin is losing its love with oxygen. Instead, the love now goes to carbon dioxide. So it releases oxygen into the respiring tissues, and then it picks up carbon dioxide. Now, what is the advantage again of this carbon dioxide behaving in that way? Automatically, you have gone through and you have got an answer. The third factor, my dear student, is body size. We have body size. Well, the oxygen dissociation curve for various animals differs according to body size. For example, the oxygen dissociation curve for small mammals is normally displaced to the partial pressure, oxygen partial pressure axis compared to the that of large animals. And this is because in small mammals, the rate of metabolism is high. And therefore, if the rate of metabolism is high, the rate of carbon dioxide production is also high. And therefore, because of increasing concentration of carbon dioxide, the partial pressure for carbon dioxide becomes also high. And at this point, hemoglobin unloads oxygen and then loads carbon dioxide. And this is important because the increase in the partial pressure of carbon dioxide causes the release of oxygen in the respiring tissues. And therefore, if you have the oxygen dissociation curve, you will see that if this is the oxygen dissociation curve, when the size of the body of the mammal is decreasing, then we have the curve being displaced towards the oxygen partial pressure axis. But maybe if this is an oxygen dissociation curve for a human being compared to the small mammal such as cat, you have the oxygen dissociation curve shifted to that particular side. A rat, which is smaller than a cat, has its oxygen dissociation curve displaced further away towards the oxygen partial pressure axis. A question: how does mammalian body sizes affect oxygen, hemoglobin binding affinity? How does the mammalian body sizes affect oxygen hemoglobin binding affinity? Now that was hemoglobin and the way it carries oxygen. Another pigment on the blood, which is also responsible for transportation of oxygen, is myoglobin. Myoglobin. This is a red pigment. It is a red pigment. Very similar in structure to one of the polypeptide chains of hemoglobin. And of course, the two proteins have been presumably evolved from a common ancestral molecule. That is, hemoglobin and myoglobin have been presumably evolved from a common ancestral molecule. Now, myoglobin, this is largely found in the skeletal muscles. It is largely found in the skeletal muscles, and this is the main reason. It is the main reason why the skeletal muscles are red. I mean, it is responsible for readiness of meat. It is responsible for readiness of meat. Well, myoglobin has a greater affinity for oxygen than hemoglobin. And for that reason, it is used when all hemoglobin has been exhausted. If the oxygen dissociation curve for hemoglobin and the myoglobin are plotted on the same axis, the oxygen dissociation curve for myoglobin appear to be displaced to the vertical axis. Appears to be displaced to the vertical axis. Is on the side of the vertical axis, while that for hemoglobin on the side of the horizontal axis. Meaning that we have two curves. Loading tension, and here we have unloading tension, 50%. This one here is an oxygen dissociation curve for hemoglobin. And here we have the oxygen dissociation curve for myoglobin, which is normally acts as an oxygen store. This acts as an oxygen store. And therefore, it releases its oxygen during strenuous body exercises. When we have heavy body exercises, when all hemoglobin has completely been exhausted, or nearly getting exhausted, that is when myoglobin comes into play. Now you can see that myoglobin is capable of loading oxygen when its tension is as low as at this particular point, but hemoglobin will never load oxygen unless the tension goes as high as this particular point. And this is indicating obviously that myoglobin has a greater affinity for oxygen because it can pick oxygen even when its tension is very, very small. But hemoglobin cannot pick oxygen unless its tension is at this particular point. My dear student, having seen the two pigments that are involved in oxygen transportation, I mean hemoglobin and the myoglobin, we are now going for a short break. We will be right back, so don't go away.